All right, welcome to chapter 3. We're going to pick up on slide number 29 and go all the way through 90. If you need to pause, take a break, you can come back to this. This will be a little bit longer. So we left off and we were talking about reaction rates. Rates equal how fast a reactant is consumed and therefore products are generated. There are various factors that are going to affect that rate. The law of mass action, aka reactant and product concentrations. Temperature is energy. An increase in temperature, therefore, is going to increase rate, and a decrease in temperature is going to decrease rate. Now, in the human body, we are not going to deal with this for the most part because our temperature tends to remain within a stable, steady zone. However, if we have hyper or hypothermia, then we would need to deal with this. Now, when it comes to hypothermia, for those of you who had med term, you'll remember hypo is going to be low and hyper is high in regards to temperature. Activation energy barrier. So how much energy do we need to input into the system in order to actually get this reaction to occur? The greater the decrease in height, the greater the increase in the rate. The greater the increase in the height, the greater the decrease in the rate. This is where enzymes are going to come to play. A lovely little chart here trying to illustrate that. All right, enzymes, as I mentioned, what role do they play? They are a catalyst. Catalysts are going to speed up the process. In this particular case, our enzymes are going to be our catalysts. They are protein chains. And we typically talk about enzymes when we're talking about digestion, so the breaking down of bonds, but they are also going to be involved with creation of things as well. They function by decreasing that activation energy for the particular reaction we're looking for. So enzymes are going to bind to a reactant or substrate. Enzyme plus a substrate is going to equal something we call ES, which is when they're bound together. And that is going to, when they separate, give us a product and the enzyme again. The enzyme is not consumed in this reaction, in this process. Now, enzymes can come in one of two different categories. They can come where they are specific, so much more of a lock and key type mechanism, which is what we have probably been taught up till now. One enzyme per compound, or they can be accessible to a group of similar substrates. So this one particular enzyme can break down the bonds of a variety of different protein chains versus just one specific type of protein chain. That's just as an example. These enzymes are not going to be changed, as I mentioned. They are not consumed. And we can always identify an enzyme by ASE, their suffix. Enzymes are always denoted that way. So if you see something like lactase or protease or peptidase, ASE, those are enzymes. If you had medical terminology, hopefully your professor mentioned that already. Just like if it ends in OSE, it typically is going to indicate a carbohydrate. Here we have a lovely diagram demonstrating for you. We have our enzyme with its active site. The active site is where the substrate can bind, and this particular active site will only accept substrate C. That's the only one that's going to fit in there. So specificity, substrate specificity, is one of two, as we mentioned, lock and key, where it's very specific only one thing can fit in. Here's the problem. Most enzymes can also be used to reverse the reaction. So if we have two amino acids that are broken apart, we can put them back into the enzyme and put them back together again, which is where the lock and key model falls short. Because of that ability to reverse, the lock and key model doesn't quite fit that, which is where the induced model comes into play. So with the induced model, it is close enough and the enzyme will mold to it. So this is demonstrating for you some of that specificity. As I mentioned, it approximates the fit, but not exactly it. As the substrate starts to bind, a conformational change in the enzyme will allow it to fit even better. Example here. Obviously, this fits better with the square, but a circular substrate comes in. It can bind to enough sites that it will then change its format to have the reaction spit out a product. And then it will go back to its original shape. Ligand protein interactions would be an example of this. Ligands are molecules that bind to protein. It's a weak hydrogen interaction, though. Or a weak hydrogen ionic interaction, I should say. There are no covalent bonds involved. It would look something like this. So don't worry about this for the test. All right. Cofactors and coenzymes. You may have heard these terms before. Ions are molecules that are required by an enzyme for the enzyme to be active. These are what we call cofactors or coenzymes. They tend to be chemical classes, and we end up with things like trace metals that tend to be cofactors: magnesium, copper, zinc, iron. As long as these metals are bound to the enzyme, the substrate can bind to the active site. And then we have vitamins, and that can transfer small chemical groups to the enzyme and act as a coenzyme. These are going to be things like niacin, riboflavin, pantothenate. Probably don't know those names, but if I were to say B3, B12, and B5, you have probably heard of those before. Those are going to be coenzymes with NADH, FAD, and coenzyme A. That's what NADH, FAD, and coenzyme A are derived from these vitamins. I can talk today. FAD and NAD, they tend to bind to two hydrogens to form FADH2 and NADH plus H. This is going to become important. These are also two energy molecules. Well, they can contribute to energy anyway. We'll talk about it. All right, factors that affect the rates of our enzyme-catalyzed reactions. First and foremost, how many molecules are generated per unit time, the enzyme's catalytic rate. Some reactions are going to generate thousands of molecules per second, while others may take more than a minute to catalyze a single molecule. So the speed at which this reaction is going to take place is still going to play a role. Just because we are changing the activation energy, it does not mean it is necessarily going to change the time it takes for the reaction to take place. This one, next one, self-explanatory, substrate concentration. If I don't have a ton of substrate in the fluid, then I cannot create a lot of product. Enzyme concentration. If I don't have a lot of enzymes, substrate can't bind. the affinity of this enzyme for the substrate. That is also going to play a role, as well as temperature and pH values again. These are just charts demonstrating some of what we just talked about. And then, just to make things even more complicated, we are going to have feedback inhibition. So just because we have X amount of substrate, just because we have X amount of enzymes, does not mean they're always going to be in play. So here we have rates of this enzyme and this enzyme are constant, and enzyme number 2 taking place is going to be dependent upon the amount of the product that is out there. So if there is a lot of product C already free floating in our extracellular fluid, then enzyme number 2 is going to become inhibited. Enzyme number 3, though, is going to continue pumping out to the best of its ability product D. So as that occurs, it's going to deplete C. As C depletes, eventually will uninhibit enzyme number 2, and enzyme number 2 will start to convert B into product C. However, over here, enzyme 1 has continued to pump out product B. So we're going to get a buildup of B. So this is used in very specific scenarios because this can, as you can imagine, be detrimental. Then we have something known as in-product inhibition. So this is where we take enzymes 1, 2, and 3 again, and they are in the process of converting substrate A into in-product D. We are going to send A through enzyme 1 to make B. B is going to go through enzyme 2 to make C. And then C is going to go through 3 to make D. If we end up with too much of product D, that is going to inhibit enzyme number 1. If we inhibit enzyme number 1, then our conversion of our substrate A into product D can't take place. So as D gets depleted, then enzyme 1 would become available again, and we can start that process. So hopefully that is making sense. All right, branched pathways. So this is an example of when we have a substrate that can become one of 2 different products depending upon the enzyme that is used. Example here, we have testosterone. If it is converted using the enzyme aromatase, we are going to get a product called estradiol. However, if it is converted by 5-alpha reductase, then we're going to get dihydrotestosterone. So it all depends upon what enzyme is used. We'll get 2 different products. ATP, you may have heard already that ATP is our energy molecule. ATP is created using exergonic reactions to synthesize an adenosine triphosphate, which is what ATP stands for. That is our energy cycle, which we'll get to here shortly. And this is what it looks like. We have an adenosine group attached to 3 phosphates. 1, 2, 3. The link between the second and third phosphate is actually the high energy bond, and when it is broken, it releases energy into its surrounding area. And then we recreate that bond by attaching it, and that's how we store energy. So ADP plus phosphate plus energy equals ATP plus water. This is going to have a net change of 7 kilocalories per mole. This is a condensation phosphorylation reaction because we're adding a phosphate group and we are getting a water out of it. If we are going to have a substrate level phosphorylation, that's going to involve an XP plus ADP. So we're going to end up with ATP plus water plus another end product. However, if we have oxidative phosphorylation, this is what happens during the electron transport chain. We have a free-floating phosphate that it can be attached to our ADP. ATP is a temporary storage. This is not meant to be permanent. All right, now let us get into how we actually get energy out of sugar. Central reaction of energy metabolism is going to be a glucose molecule. It is a C6H12O6 glucose molecule plus 6 oxygens, and that is going to give us an end count of 6 carbon dioxides, 6 water molecules, and a whole slew of energy molecules, with a net change of negative 686 kilocalories per mole. Give off a ton of energy. So here we go. Glucose oxidation gives off heat and energy. ADP is added to a phosphate group to create ATP. Those ATP bonds are going to break, giving off energy and heat again, and this is going to allow that energy to be then used for work. Cellular movement, molecular synthesis, transport across membranes, whatever it may be. That gives us an ADP and phosphate free floating again, and we just rinse and repeat. Because this ATP is a temporary storage vehicle, we cannot create a ton of ATP in one cell and then transport it to another cell to actually be used. This is why every cell in our body is going to have mitochondria, and the ones that need a ton of energy, think brain cells, muscle cells, they're going to have a ton of mitochondria to make that energy. So, if we are doing this conversion of glucose in the presence of oxygen, so we have our 6 O2 molecules, we also need to have 38 free-floating ADP and 38 free-floating phosphate groups. This is because one glucose molecule in the presence of oxygen will create 38 energy molecules, 38 ATP. So, efficiency of this conversion process is about 38.8%. The vast majority of the energy created during all of these chemical breakdowns is given off in the form of body heat. So this is why we are warm. Glucose oxidation has multiple steps, and to be perfectly honest, most of the time you will see textbooks refer to 3 steps. Now, the reason for this is because that oxidative phosphorylation step is usually combined either into the Krebs cycle or the electron transport chain. However, our textbook is separating it out, so that is what we are going to do. Step number 1. This step happens to take place within the cytosol or cytoplasm of the cell. So this can take place without any mitochondria being around, can also take place without the presence of oxygen because no oxygen is consumed in the making of the end products of this particular step. This particular step is going to involve a sequence of 10 enzyme catalyzed reactions that will convert one glucose molecule into 2 pyruvate molecules. This is going to gain gain us a net gain of 2 of our 38 ATP molecules, and 2 NAD plus molecules are going to become 2 NADHs. We're going to give up an electron here. We do not produce CO2 and we do not produce oxygen. This is what that would look like. So this is the same exact thing. It is showing the same exact thing. It is just showing it. Sorry, no, it is not. This is the continuation, my bad, of the glycolysis. Is just showing it in two different formats. Showing you here the ring structure, and then we come out of it. Do you need to memorize this every single step? Not for this class. If you happen to take a biochemistry class, you will have to memorize the major steps and the major enzymes involved. But glycolysis, 2 ATP and 2 pyruvates are created from 1 glucose molecule, no oxygen necessary. And I'm sorry, my mic doing this at school is probably picking up everything. And this is just showing it another way. All right. Now, these 2 pyruvates do not immediately go into the next step, which is the Krebs cycle, otherwise known as the citric acid cycle, depending on what textbook you're looking at. There is something called a linking step. And this linking step is going to convert the pyruvate molecule into an acetyl-CoA molecule. And it's going to do that for each of the pyruvates. Each acetyl-CoA molecule will then go into the Krebs cycle. Again, we are not going to require oxygen. However, we are going to give off carbon dioxide, as well as a NADH and a hydrogen ion. So the NADH that was consumed in the glycolysis step, we get back. This is a unidirectional step, meaning it is a one-way valve. Once we go into the Krebs cycle, this is cyclical. The acetyl-CoA is always the start of this cycle. It's our initial substrate. And we reduce coenzymes in various steps to get our energy and a whole bunch of other byproducts. The Krebs cycle is where if we are using proteins or fatty acids for energy, they are going to go into the cycle here. And this is what it looks like. Here we have our acetyl-CoA. We also insert a water right here, combine it with citrate synthase. Coenzyme A is given off. We get citrate. We then use a conitase, which is going to convert it into isocitrate. Then we use isocitrate dehydrogenase, and that is going to give us an NADH plus hydrogen plus CO2. And that's going to form alpha ketoglutarate. We're going to insert coenzyme A back in along with an alpha ketoglutarate dehydrogenase, and that will give off a NADH plus H plus and another carbon dioxide, making it succinyl-CoA. Okay, now we put in a phosphate and a water molecule along with our succinyl-CoA synthase, and it will give off a GTP, which will then convert an ADP into an ATP, and we get our CoA back. That creates succinate. Succinate dehydrogenase will convert that into fumarate, giving off a FADH2 in the process. We're going to add in a water and fumarase, and that will give us malate. Sorry, water. Malate, we're then going to have malate dehydrogenase, and that will convert it to oxaloacetate and give us another NADH plus H plus. We'll add in our water, and we are back to where we started. You see how this goes round and round and round and round. And there are a variety of factors that can put a halt to this process. The big one being the presence or lack of presence of things like oxygen. Krebs cycle takes place within the mitochondria. Again, do you need to memorize every single one of these steps? No, not for this class. However, biochem, you will. Obviously, that depends upon your teacher, but most biochem teachers have you memorize that. And then have to spit it back out. All right. Translation of all of that. Acetyl-CoA is our initial substrate. The production per acetyl-CoA is going to give us 1 GTP, which is going to produce 1 ATP, and 3 NADH, 3 hydrogen ions, and 1 FADH2. As a waste product, we're going to get 2 carbon dioxides, and this is per acetyl-CoA. Remember, each glucose created 2 acetyl-CoAs, so this is times 2. So, end of the cycle, with the presence of oxygen, 1 glucose creates 38 ATP. We have now, so far, created 4 ATP, 2 in our glycolysis stage and 2 in our Krebs cycle, 1 per acetyl-CoA. All right. From here, we're going to go to oxidative phosphorylation. This is going to bring us to our electron transport chain, which is where the vast majority of our ATP is going to be created. Again, this is going to take place within the mitochondria, and this is the step that absolutely requires the presence of oxygen. The electron transport chain is called such because electrons move down a chain that is used to synthesize ATP. This is a chain of molecules in the inner mitochondrial membrane. So it's folds. Molecules are going to go undergo oxidation reduction reactions, going from high to low energy as the electrons are passed along them. Energy is going to be released in this process, and that is going to be used to synthesize ATP using our oxidative phosphorylation and ATP synthase. And the last electron acceptor within that chain is a oxygen. And this is why the presence of oxygen is required. So it looks something like this. We have an NADH plus coming in from our glycolysis linking step in Krebs cycle. That's going to come in at the beginning of the chain, and the hydrogen molecules are going to come off. We're going to get just a plain old NAD plus. So we'll get 2 free floating electrons and our 2 H pluses. They're going to come into this step, and between each step, we're going to get an ADP plus phosphate and an ATP. It's just going to happen over and over and over again as those electrons and hydrogens bounce down the chain. Coming in from the Krebs cycle at about coenzyme Q, we are going to get our FADH2. It will lose its hydrogens and 2 electrons to become FAD, and it too will then go, though its electrons and hydrogens will go bouncing down the chain until it gets to the end, where we are going to get ATP and water molecules. Net reaction. Every NADH is going to create on average 2.5 to 3 ATP per oxygen molecule. Just straight up oxygen, 1 oxygen, not O2. And every FADH2 is going to create 1.5 to 2. The reason for this variance is because the exact number of ATP is going to depend upon the condition of the cell. And it would look something like this. So remember, we have our mitochondria, we have our cristae, which is this folded inner membrane. Located within that is its matrix. And passing back and forth along that cristae is where this electron transport chain is going to take place. So this is where everything happens here. From our glycolysis linking step in Krebs cycle to FADH2, 10 NADH plus H pluses are going to come into the oxidative phosphorylation along with 6 O2 molecules or 12 oxygen atoms, 28 ADP and 34 phosphates, and it will end up spitting out 10 AD pluses, 2 FADs, 12 water molecules, and 28 ATP. And this is how you put it all together. Glycolysis, linking step, Krebs cycle, or citric acid cycle, depending on what textbook you're looking at, electron transport chain / oxidative phosphorylation. 6 carbons, or carbon dioxides, 6 water molecules, and 32 ATP total. All right, what about if we don't have oxygen? Well, then we are going to have low energy, very low energy. So, low oxygen availability, the electron transport chain is going to back up. Because it backs up, the Krebs cycle is going to stop. Glycolysis will continue as long as we still have NADHs available. And as a byproduct, instead of getting acetyl-CoA, you will get lactate. 1 second. I cannot write and talk at the same time. So here's what we do. Glycolysis, pyruvate, lactate dehydrogenase is going to come into play instead to continue to give us NADH. And we are going to then have lactate, NAD+, sorry. Conversion of pyruvate to lactate. This allows our muscles to operate with low oxygen. As we've seen, very inefficient. It's about 2% efficiency. That lactate is going to accumulate, and that is going to be disruptive and create what we call an oxygen debt. When oxygen levels return, that lactate is going to be converted back to pyruvate, so it can then restart the Krebs cycle. However, what's first going to happen is the oxidative phosphorylation is going to resume first. Sorry for the kids outside. It's going to resume first. This is the oxygen debt. We have to have enough oxygen in play to get rid of the backup on the electron transport chain and get rid of the lactate. Once the lactate has been converted into coenzyme acetyl-CoA, and that backup has been taken care of, the oxygen debt has been forgiven. All right. Energy use, storage, and use. Carbs, lipids, proteins. We've already talked about carbs. Glycolysis. Our proteins are going to get broken down into amino acids. The nitrate group is going to be removed, and that is going to form urea. And various products can come in at the pyruvate stage and be converted into acetyl-CoA. Some can come in straight at acetyl-CoA, depending on what we are looking at here, and some will get converted into this step on this side of the Krebs cycle. Triglycerides, on the other hand, the glycerol group is actually going to come into the glycolysis stage The fatty acids are going to be converted into acetyl-CoA, as well as coming directly before oxidative phosphorylation. You might be thinking about how we store glucose. It gets stored in the form of glycogen. And simply this, we use hexokinase to convert it into glucose 6-phosphate, which can then go into the glycolysis and become pyruvate. Glucose 6-phosphate, which is one of the intermediate steps in the glycolysis cycle, can be converted into glucose using glucose 6-phosphatase, but it can also become glycogen. I might have just misspoke. Hexokinase converts glucose into glucose 6-phosphate, different cycle to become glycogen. Yeah, I did misspeak. Okay, glycogen metabolism. Glycogen is stored around our liver and kidneys, and it is going to be quick energy and is transported to other cells via your blood. The muscles, skeletal muscles, also contain some glycogen stores around them, but not nearly as much, so we don't tend to talk about it. The glucose can leave the cell, but not glucose 6-phosphate, which is why we are going to convert into glucose if we need to transport it out of the cell. Only the liver and kidney, though, are going to have glucose 6-phosphatase, so only the liver and kidney can convert glucose 6-phosphate into glucose. Gluconeogenesis. This is going to convert glycerol, lactate, and certain amino acid chunks into glucose. Again, mainly going to occur in the liver. This is why the liver is heavily taxed if you're on something like a keto diet. It is working hard. This is why your liver is also heavily taxed if you are do any strenuous workouts that lactate. This is not simply the reverse of glycolysis. There are a whole slew of other enzymes involved, and we're not going to get into that. But it will look something like this. Amino acids, urea comes off, keto acids can go straight into the Krebs cycle in certain spots or become pyruvate, and we can reverse it into glucose, which can then be sent out or sent into the Krebs cycle. Well, glycolysis and then into the Krebs cycle. Energy storage, as we have mentioned in passing, energy is stored in the form of triglycerides, which are 3 fatty acids joined to a glycerol. Lipids are going to have a greater energy per weight than carbs or proteins, and that is due to those carbon chains. And most triglycerides are going to be found stored within our adipocytes. And for those who have had med term, you recognize adipo as the root for fat. So our fat cells. All right. To convert that triglyceride into energy, we need to undergo the lipolysis, which is the breakdown of those triglycerides into energy. We've got to remove the glycerol from the fatty acids and then chunk the fatty acids into carbon bite-sized pieces. Glycerol goes into glycolysis, becomes pyruvate. Fatty acids can become, with a little bit of modification, acetyl-CoA. That process, though, is going to give off ketones, which is why if you are using fatty acids as your primary fuel source, you're going to end up with a sweet and fruity breath. Ketone bodies smell sweet and fruity, and your body is going to be getting rid of them however it can. And then so on and so forth. That breakdown is going to occur with lipase. Lipase is going to separate the glycerol from the fatty acids. Eventually, that glycerol will become dihydroxyacetone phosphate, which will go into glycolysis. Glycerols give off 20 energy, 20 ATP. The fatty acids are going to undergo beta oxidation, 2 carbons at a time. So every 2 carbons becomes an acetyl-CoA with an NADH and a FADH, which will go into the Krebs cycle and the electron transport chain respectively, which means every 2 carbons will create 17 ATP. So the longer the carbon chain, the more energy created. This is also why fatty acids, or triglycerides, the tails, are always in even numbers. You're not going to find a 17 carbon chain. It's always even numbers. One of the reasons why, anyway. And that looks something like this. Wait, no, this is our amino acid again. Why is this here? Because we're going into amino acids again. Proteolysis is going to be our breakdown of our proteins chain into individual amino acids. At that point, we're going to have deamination take place, which is when the amino group, that nitrate group, is removed. That will undergo its own process and become keto acid plus ammonia, NaH3. Those are waste products. The keto acid, though, is going to become pyruvate or acetyl-CoA and go into the Krebs cycle. The ammonia is the byproduct there. And that is the end of chapter 3. Sorry for the background noise. Make sure you take breaks, and we will see you in class.